Hello everyone, I am Anindita and I'm a PhD student from Bioscience and Bioengineering Department at the Bombay. My supervisor for this project will be Professor Hari Verma, and I'll be working on this project named a stochastic partial differential equation model for laser speckle-based cerebral blood flow imaging. Now, there are for understanding the cellular metabolism, whether is it is normal or not. There are two um, quantities that we are measuring. One is oxygen consumption and supply, and second one is the blood flow. There are several techniques which measure these two quantity to understand the physiological conditions of the animals. We are generally opting for the optical methods as it is a non-invasive techniques. Now, when light propagates through the tissues, as it's a turbid media, it attenuates, and there are two tissue optical properties we get from this attenuated light. One is mu A and mu S dash, optical absorption coefficient and reduced optical scattering coefficient. The photons are being modeled by this radiative transfer equation, which has several applications in physics and engineering. Now, we generally try to see our interest is the NIR light, as in this region the other chromophores are absorption coefficients are low, so we get the more scattered light, which has more information about the tissue. Now, there are several existing methods that we are doing in our lab. One is DOS, LSCI, and DCS. These have some limitations. For example, LSCI provide relative blood flow images, and it is sensitive to the illumination and motion artifacts and cannot directly measure velocity, whereas DCS actually provides precise modeling, and it has low SNR in deep tissue and also assumes stationarity. Both methods struggle to capture the non-stationarity blood flow of the blood flow dynamics and suffer the data loss at the low sampling rate. Now, DOS is modeled by generally photon diffusion equation. Laser speckle contrast imaging takes account of the speckle, measure the speckle contrast, and DCS actually takes care of the intensity autocorrelation. This is a normal speckle, and this is how a speckle looks like. This is the experimental setup of a LSCI, and this is the experimental setup of the DCS. Now, we try to formulate the correlation diffusion equation as a stochastic partial differential equation, so that it can take account of the stochasticity of the blood flow. This noise also can be included, and we can also solve it through these numerical methods and the machine learning. So we can verify our method by the numerical solutions such as FEM and Monte Carlo based method, and also machine learning algorithms can be used. Also, very experimental validations can be done using these. Now, there are our proposed methodology actually takes account of the accuracy, robustness, and real time inference, and it also helps to helps for the brain injury and stroke monitoring, and it has application for the clinical translation also, and it also improves the light propagation in broader case, light propagation in the fog or the turbid medium, and gives better visualization for complex environment. These are the these are the references, and thank you so much for the attention.